Good evening, everybody. Welcome to week 11 of the problem solving session. We will be starting in uh, 5 to 10 minutes so that everybody can join in. We'll wait for a couple of minutes and see if anybody joins in, and then we'll start. Okay, a very warm welcome to all of you. Good evening. Welcome to the 11th session of the problem solving interactive sessions for the course Genome Editing and Engineering, which is taken by Professor Utpal Bora of IIT Guwahati. I am Sanket Mukherjee, Prime Minister's Research Fellow from the School of Biological Sciences at IIT Delhi. So let's get to the questions first. So the theme of this week was personalized medicine. So let's see what do we have for the questions. So the first question is Ayurveda classifies all individuals into different prakriti based on the theory of A. Vata, B. Pitta, C. Kapha, or D. Tridosha. I'll give you a minute to think about it and then we'll discuss the answer. Okay, so the answer to this question would be Ayurveda classifies all individuals into different prakriti based on the theory of three dosha. D. So let's talk about Ayurveda. That was one of the first beginning of personalized medicine. So Ayurveda is a natural healthcare system that dates back more than 5,000 years in India. It thinks that each individual is unique and has a particular constitution and hence emphasizes on personalized treatment. Ayurveda classifies all individuals into different prakriti based on the theory of three dosha. That gives us the answer. This is independent of racial, ethnic, or geographical considerations and may provide appropriate means of classifying phenotypes to be considered collectively for genotyping. So, if we talk about the history of personalized medicine, space, air, fire, water, and earth, these are the five essential components of life that exist in human body as the three primary humors known as the three doshas. So they are Vata, Pitta, and Kapha. So among the three doshas, these are the three components, three essential uh, components which are very important. So the component of space and air combine to form the Vata dosha, right? Uh, Pitta dosh is made up of the elements of fire and water, and Kapha dosh is derived from earth and water components. So this is the basis of Ayurvedic medicine, and it is not dependent on any of the geographical factors, ethnic factors, racial factors, and so on. So let's go to the next question. Ayurveda categorizes drugs according to Ras Panchaka, Ayurvedic pharmacology, which states that the drug's action is attributed to certain factors present in the drug as follows. Find the wrong combination. Is it A, rasa or taste, B, guna or effect, C, virya or potency, or D, vipaka or post-digestive taste? So I'll give you a minute to think on this and then we'll discuss the answer. Okay, so the answer to this question would be that Ayurveda categorizes drugs according to the Ras Panchaka or Ayurvedic pharmacology, which states that the drug's action is attributed to certain factors present in the drug as follows. The wrong combination among them is guna effect. So let's see about Ayurveda. So on continuing the history of the personalized medicine, Ayurveda, it categorizes drugs according to the Ras Panchaka or Ayurvedic pharmacology, which states that the drug's action is attributed to certain factors present in the drug, namely the ras or the taste, gun or the property, virya or the potency, vipaka or the post-digestive taste, and the prabhav or the effect. Whereas modern pharmacology attributes the drug action to the chemical structure of the molecule. So the Ras Panchaka modality can deliver treatment because it considers the person's prakriti as well as the pharmacodynamics and pharmacokinetic properties of a drug, as opposed to modern treatment, which elicits different responses from person to person, even when using the same drug for the same disease. So, since Ayurvedic pharmacology it uses a lot more factors than the modern medicine, which only considers the structure of the drug uh, and some properties, physicochemical properties of the drug. Apart from that, there is nothing that is taken into consideration in modern medicine. While Ayurveda also considers the prakriti of the person or the patient rather. So this is the reason why Ayurveda is believed to be more effective and since the therapy is personalized based, even the diagnosis is personalized based on the state of the patient, which varies from individual to individual, even when uh, there is uh, uh, the same disease that happens. So that's the idea. Now let's come to question number 3. Garrod is best known for his book Inborn Errors of Metabolism, where he claimed that 4 diseases, alkaptonuria, albinism, cystinuria, and pentosuria, were inherited as Mendelian autosomal recessive trait and was published in the year 1909, 1902, 1809, or 1990. So I'll give you a couple of minutes to think about it, and then we will be discussing the answer. Okay, so Garrett is best known for his book Inborn Errors of Metabolism, where he claimed that four diseases, alkaptonuria, albinism, cystinuria, and pentosuria, were inherited as Mendelian autosomal recessive trait and was published in the year 1909. So let's see what we have. So Archibald E. Garrett and his thoughts on personalized medicine. So in 1902, Garrett published the paper "The Incidence of Alkaptonuria: A Study in Chemical Individuality," where he suggested that alkaptonuria is not the manifestation of a disease, but is rather of the nature of an alternative course of metabolism. He is best known for his book, Inborn Errors of Metabolism, published in the year 1909, where he claimed that four diseases, alkaptonuria, albinism, cystinuria, and pentosuria, were inherited as Mendelian autosomal recessive trait. This foresightful effort paved the way for the study of hereditary illnesses and established Garrod's status as the father of medical genetics, that is biochemical and molecular genetics. So he basically gave the idea that diseases also have a biochemical and molecular mechanism involved behind them. So that's a very important thing that we should consider. So if we see the timeline of modern medicine, how it has evolved over the last, I mean, over humanity. So in 8000 BCs and before that time, there was prehistoric medicine. Then there was ancient Egyptian medicine. Ancient Chinese medicine was at around 2700 BC. Babylonian medicine was at around 2300 BC. Ancient Indian medicine was at around 1500 BC. A very important timeline. Greek and Roman medicines, they came up at 1200 BC to 300 AD, and 16th to 17th century was the time for Arabic medicines. And from 20th century till now, and even in the future, we have the modern medicine, which is still evolving, and we are finding out a lot of things. But it is very intriguing to know that a lot of things that we are discovering through science in today's generation, today's era, has been inspired, or I'll not say inspired, rather it has been documented in some of the ancient texts, at least in Indian medicine and in some other forms of medicines as well. So it is very intriguing to know how do people at that time, that era, with that much less technological advancements, were able to find solutions to problems which are even relevant now. So that is a question that people do ask. And but anyway, like for example, personalized medicine was discovered or discussed in Ayurveda as well. So that's very interesting to find that out. So let's go to the next question. In 1926, Pamaquine and 8-aminoquinoline was one of the most effective medications used to treat A, AIDS, B, cancer, C, dysentery, or D, acute malaria. I'll give you a couple of minutes to think on this, and then we'll discuss the answer. Okay, so in 1926, Pamaquine, an 8-aminoquinoline, was one of the most effective medications used to treat the acute malaria. So let's see. Usually quinoline kind of compounds are used for malaria. Let's see. So treatment of malaria was one of the beginnings of personalized medicine. So malaria was first recorded in ancient Chinese medical archives in 2700 BC. Even today, malaria is considered as one of the most serious and lethal disease. Several plants, including Qinghai in the 2nd century BC in China and the cinchona tree in the 16th century in Peru, have been used to cure malaria. So what are the symptoms? There is a headache. Uh, there's a systemic fever. There's muscular fatigue and pain. There's back pain as well. In stomach, there is nausea and vomiting. In spleen, there's a splenomegaly, basically, during malaria. There is respiratory problems like development of dry cough. And in the skin, there is chill that comes from the fever and also excessive sweating. So these are some of the symptoms of malaria. So how was it treated? So Pamaquine, an 8-aminoquinoline, was one of the most effective medications used to treat acute malaria in 1926. However, administration of Pamaquine showed the adverse effect of hemolytic anemia in many patients. As a result, scientists began researching alternate therapy to combat the side effects of Pamaquine. So let's see what happened. So, in for treatment of malaria in the Korean War, that was around 1945. Uh, Primaquine and 8,4-amino 1-methyl buta 1-amino 6-methoxyquinoline, which is also a quinoline derivative, was used as an anti-malarial drug to eliminate the long latency of Plasmodium vivax infections of the soldiers. So if you see the infection cycle of Plasmodium vivax, which causes malaria, there's a long latency period involved. So because of this, this drug was used. In 1956, Carson et al. Discovered that the side effects of hemolytic anemia were caused by a deficiency in glucose 6-phosphate dehydrogenase enzyme. It's a very important enzyme in glycolysis. So it's involved there. So that is the reason which why hemolytic anemia was caused. The discovery of the connection between anti-malarial drugs and G6PD deficiency led to a new perspective on the adverse effects of these drugs, as well as a more personalized approach towards the disease, which was one of the first examples that led to a big step towards the application of more personalized therapy. So, so it was during that time when people realized that a particular drug, if you intend to make it work at a particular location for to a particular protein, it might also affect some other processes, metabolism of the body. It may hamper it, it may destabilize it. So to treat diseases not only based on symptoms, but also on the molecular basis of the disease, how they are, they are caused and how they are, they progress, all these things need to be considered. So in 1550 BC, there was Egyptian personalized medicine. Around 2700 BC, the epidemic of malaria had come into picture. So it's a very old disease, yet it is dreadful even today. So around 500 to 400 BC, around 510 BC, this G6PD deficiency was found out. So the Hippocratic personalized medicine was there. Then there was a gap in this. In 1950s, the DNA structure was found. So you need to realize that these kind of problems were much before even DNA was considered as one of the genetic elements which causes or which is the code of life. Even before that, people had disease and they were treated. So then biomarkers were introduced, the different families of biomarkers and receptors were discovered. And then understanding the genetic code, it begins after the discovery of DNA. So it all happened in the 20th century, late 20th century, where people tried to understand how DNA play a role in various diseases. So after a decade or so, or maybe after a couple of decades after DNA was understood, the term terminology of personalized medicine, it appears. So because from now, a lot of key players that are involved behind the disease were identified, and their role, the way they function and everything, those things were considered. So a HapMap project was done in 2002. GWAS, first GWAS was published in 2005, and there were plenty new discoveries right from the early 21st century. So the epidemic of COVID-19 that happened in the late 2019 also accelerated the need of personalized medicine. So because we try to understand, because even for COVID-19, the effect of various factors, because the disease, while the virus is same, it caused a different kind of severity of disease in different patients. So we came across comorbidities, we came across other issues, we also came across a lot of heart problems, that cardiovascular diseases that have come up due to COVID. So this very reason led us to implement personalized medicine, to have accurate diagnostic tools, to understand where the problem lies, and to cure it properly. So this is why at this time personalized medicine is so important. So let's go to the next question. Question number 5. Shendure's group published the first proof of concept that NGS technology, next generation sequencing technology, may be used to diagnose genetic diseases in September 2009. After few months, they reported Miller syndrome, the first recessive disorder, through A, whole exome sequencing, B, whole genome sequencing, C, whole proteome sequencing, or D, whole plasmid sequencing. So I'll give you a couple of minutes to think on this, and then we'll discuss the answer. Okay, so Shendure's group, they published the first proof of concept that next generation sequencing technology may be used to diagnose genetic diseases in September 2009. After a few months, they reported Miller syndrome, the first recessive disorder through whole exome sequencing. Although the rest of the things, they just mean that. So exomes, if they are sequenced, then it's whole exome sequencing. Whole genome sequencing, it includes the exons and the introns, whereas proteome sequencing only involves the proteome and the proteins basically, and whole plasmid sequencing, it is not relevant in this context. So let's see, let's discuss a little bit about next generation sequencing and its implications on personalized medicine. So next generation sequencing, it has helped in the development of personalized medicine by providing scientists with a strong tool to comprehend each patient's condition and its unique genetic features and the whole genome mutational status with single nucleotide resolution. The Human Genome Project, it completed in 2001 and opened the door to a better knowledge of precision medicine. So what essentially is the idea here is that cancer patients, they will be having cancer, they will be having tumors, which are probably malignant tumors. So now these cells will be sequenced, the cancerous cells will be sequenced, and then we will analyze what are the mutations in these cells, in these specific types of cells, which is leading to the disease. So that's where personalized therapy comes into picture, because if you know the root cause of the disease, where what particular mutation has brought up that um, disease, then we can definitely revert back that mutation to the wild type, and probably the disease will no longer be there and there would be no adverse side effects. For that, we can use tools like for genome editing, like the CRISPR-Cas9 technology or zinc finger nucleases or talins, things like that. So yeah. So if we talk about the evolution of next generation sequencing and personalized medicine, so in the very beginning in 1977, the Sanger's method of sequencing was developed. Till date, it is one of the most preferred method of sequencing. Still, it is used. That was when the sequencing game started. In 1981, Human Mitochondrial Genome Project was done. And human mitochondrial genome is relatively smaller in size, although it also has a huge size, because since it has an endosymbiotic origin, it is similar to prokaryotes. The genome size is smaller. So it was first discovered, or it was we started sequencing the mitochondrial genome first, and then human genome was sequenced. So after that, in 1990s, it was done, and it took a lot of time, almost more than a decade. It took that much amount of time because we were dealing with a lot of data and we did not have um, appropriate computational power to analyze or process or even store that much amount of data. So that was one of the issues. And then in 1995, the complete cell genome, again, with respect to prokaryotes, this was done. In 1996, we started with eukaryotic genomes of lower order organisms. However, all during this time, the whole Human Genome Project was still active. In 2001, the Human Genome Project was completed. And it was around 2005 that we had the second generation sequencing. So second generation sequencing is a little bit different. That involves a little different, processes. And then we had the second gen sequence analyzer for analyzing the sequence data. And then there was research on human microbiome project. So since we are now getting better in sequencing, we have we can analyze the variety of microbiome that is present in humans, and that is it is very important in a sense that to understand that the microbes or the microbiota of humans also dictate the physical state of humans, the disease state of humans, and the prognosis basically. So that was a very important discovery. Then the third generation sequencing like PacBio came up at around 2011. In 2014, nanopore sequencing came up by Oxford Technologies. So that was again another kind of sequencing technology. Now the size of this entire machine is going down and the computational power is going up. So from 2019 onwards, there was the beginning of the third stage human microbiome project. And from the beginning of SARS-CoV-2 epidemic, pandemic rather, there were extensive sequences of viruses, which comes under microbiome, which was done. So SARS-CoV-2 is one of the only viruses that have millions and millions of data sequences available in database. So uh, this sequencing technology, the evolution of sequencing technology is not only important to analyze how diseases, I mean, how the human body reacts to certain diseases, but also the evolution of viruses, bacteria, pathogens in general, how they evolve over time and what is their response to antibiotics or antivirals or even vaccines as a matter of fact. So these kind of research has come up and even it is going with full swing nowadays for viruses as well, for bacteria as well, for fungi, for a lot of things basically. So the entire biology has entered into the sequencing race essentially. So after this, let's see how next generation sequencing library is prepared. So essentially you have the genome. So to get the genome, you need to isolate the genome from the cells. That's the first idea. Then you will be performing DNA fragmentation. You need to have these genomes into smaller sizes, and you will add adapter molecules at the end of each of these fragments. It's very important because adapters would be the area from where the initial, you know, uh, if in case of PacBio sequencing, there is a fluorescent readout. So that's where the primers would bind, and then you'll get the sequence of the rest of the your region of interest. So this is very important in that sense. So after the library preparation, what we do is the library hybridization. So now that we have these, uh, uh, the adapter molecules attached to your target DNA of interest, now you would amplify this DNA molecule. That's the idea because they will have these adapters. So you will amplify them using a normal PCR reaction. Then you will perform cDNA library sequencing. So during all this process, you'll have a cDNA. If you're starting from RNA, then you'll have a cDNA. Now, the sequencing of this cDNA will be done. As I said, in case of pyrosequencing, we use fluorescent labels, and there will be multiple sequencing cycles. And due to this process, what we will have is sequencing data. Now, this sequencing data will be referred to a reference sequence. That is the sequence that you know is of the organism. So that would be the reference sequence. And to this reference sequence, you will align these reads, which are different regions of these references. And after alignment, you will get something called, you'll get the variant data. You'll get the annotation of variant data. Where is the variation present among compared to the reference, where is the variation? So that kind of things can be done. It is also important to note that for de novo sequencing also, NGS can be used for sequences where references are not known. Like for them also, these things can be prepared, but it becomes a little bit difficult, and the validation is it's a little bit tougher in that situation, because the algorithms need to work on that situation. So there are few NGS limitations also, like it does not work very well with repeats. Um, and many other things. So that's not the point of the discussion. But the thing is, NGS is the next step towards personalized medicine. So the steps involved in next generation sequencing is first the library preparation, and then the amplification, PCR amplification, similar to that, and then sequencing and data analysis. And where data analysis is one of the major, challenges in NGS data, and that has introduced a lot of data science and big data concepts in biology. The data analysis involved base calling, reads, alignment, variant identification, variant not annotations. So yeah. So talking about the implications of next generation sequencing in personalized medicine. So the key concerns of medical diagnosis is to identify the genes and mutations responsible for human disorders. Early diagnosis of genetic abnormalities, carrier status, and genetic predispositions to cancer and cardiovascular disease may minimize healthcare cost and disease severity. Shendure's group, they published the first proof of concept that NGS technology may be used to diagnose genetic disease in September 2009. After a few months, they reported the Miller syndrome, the first recessive disorder through whole exome sequencing. So that gives us the answer. So if you want to know how the role of NGS in developing a personalized medicine, so when you get the genomic data from a patient, for example, or from a human, and there are a lot of companies, especially in the US, that do this work. So they do sequence your genome. So you have to send them samples, and it's around $1,000 which they take for each sample, and they will essentially isolate your DNA, sequence them, and will tell you what are the uh, problems that you are having, or what are your predispositions to certain diseases, which are genetic diseases. So that could be done. So physical examination, transcriptomic, proteomic, and metabolomic analysis, family history can also be found out. So ancestral analysis, pedigrees could be also traced through this kind of technology. So bioinformatic analysis and interpretation could be done, and based on all these data that we have, we can go for treatment. So regimes. So that's the idea. So in the figure here, you can see the personalized treatment based on comprehensive NGS-based genotypic data. So in a traditional medicine, most diseases are treated according to the clinical characteristics without taking account of the patient's genotype. Although these approaches are effective and secure for the majority of patients, some may not respond to the prescribed drug or may encounter negative side effects. Next generation sequencing can be used to collect genetic information, to identify the outlier patients in advance, and to suggest potential alternatives for them. The NGS data of the particular patient are used to identify variation in the genes encoding proteins involved in drug absorption, distribution, metabolism, and excretion, basically the ADME characteristics of a drug, and therapeutic targets, as well as the regulatory areas. All pharmacogenes activities ratings are calculated based on the combined effects of all detected variations. This might These might offer recommendations to responsible doctor on the medicine selection and dosage. So while there are certain, in traditional medicine, which is still used because it's at least cheaper, So on that effect, in clinical trials, the traditional medicine, it fares pretty well among majority of people, but we do not take account of a lot of other things, a lot of genotypic differences that might arise. A lot of In a lot of situations, the medicines are only effective for specific races or ethnicities as well, because there are differences in the genome based on these features as well. So these kind of problems can be solved using next generation sequencing. However, the technology itself is very new, and it's expensive, not only computationally but also um, the skilled force that you will be required to analyze NGS data, to have them, store them, analyze them, and to make some meaning out of them. That requires a lot of understanding. That requires a lot of research. So, for example, there are many single nucleotide polymorphisms which are already known in humans, but what are their implications? That is not known. Not all of them are known. So these kind of issues do come up. So a lot of research is still pending, and this is a lot of data, mind you. So for these kind of problems, we need more amount of people who would get into this. So let's see what are the limitations. So although NGS is an useful tool for studying genomic alterations that connect to clinical diseases, it has several limitations, such as analytic sensitivity of mutation detection, which makes it difficult to identify a low tumor proportion and lower mutation owing to tumor heterogeneity. In NGS systems such as Illumina, systemic and sequencing mistakes are prevalent. Current NGS technologies are unreliable when it comes to recognizing homologous genes, GC-rich regions, and repetitive regions. Another big challenge is the interpretation of NGS data, and the databases may always not be reliable because structural and copy number variation necessitate independent bioinformatic algorithms. Numerous approaches must be coupled to read the NGS analysis. So while we have a lot of data which is generated from next generation sequencing, but to analyze those data and to develop algorithms based on those data, though that is still a challenge. And also in case of tumors, there's tumor heterogeneity. So there are multiple mutations among different cells, few of which are very rare. But these few mutations, when you try to target the tumor, maybe the tumor population, which was majority in number, they might die, but these very rare mutations might have given them the ability to survive, and those come up. So that causes a lot of remission situation in case of cancers, and those kind of mutations which are very, very infrequent in number, catching those mutations becomes difficult because there is a error rate also that you have to take into account. So that kind of situation still pose a problem for NGS, but definitely better algorithms would help uh, us to identify these noises and to remove them effectively. So yeah, more research is required. Uh, So that was a pretty long discussion on next generation sequencing. We'll go to the next question. That is question number 6. Engineering smart biomaterials for drug delivery systems that can detect the demands of a person's physiological condition and change their drug release profile accordingly is an appealing method for creating a precision immunization, B precision pharmacokinetics, C pluripotent stem cells, or D precision biomaterials. So I'll give you a couple of minutes to think on this, and then we'll discuss the answer. Okay. So engineering smart biomaterials for drug delivery systems that can detect the demands of a person's physiological condition and change their drug release profile accordingly is an appealing method for creating precision pharmacokinetics. So the very fact that the drug loading, drug release profiles, and the kinetics involved is pharmacokinetics. So let's see what precision pharmacokinetics is. So for understanding this, we need to know the engineering biomaterials for precision medicine. So advances in biomaterials research has enabled us to make creative product designs to target and cure diverse ailments um, may be required to meet the rising need of for precision medicine. So precision biomaterials are engineered with specific mechanical and biochemical features to enable the design of personalized implants or organ replacements. Precision biomaterials must be designed to perform a variety of unit operations, such as separators, which can specifically attach to molecules or cells for separation, sensors which can detect particular analytes or electrochemical signals. Then there is responders which can change their morphological features or degrade, and controllers which can change the material's properties to release payload. So these all things are there. Now about on engineering the biomaterials, in addition to supporting the growth of artificial tissues, biomaterials can be specifically designed for direct in vivo tissue manipulations as well. Biomaterials could help in enhancing the efficacy of precision medicine through the engineering of smart drug delivery systems. Through pharmacokinetics, the drug delivery systems may foster the accuracy of precision medicine. So there are certain polymeric biomaterials, also nanomaterials, as a matter of fact, which are used for drug delivery, skin tissue engineering, bone tissue engineering, cartilage tissue engineering, corneal tissue engineering, and cardiac tissue engineering, some of the very basic things. So engineering smart biomaterials for drug delivery systems that can detect the demand of a person's physiological condition and change their drug release profile accordingly is an appealing method for creating precision pharmacokinetics. That answers the question. So among the biomaterials that are used, they're nanomaterials or particle-based drug delivery system is a very promising approach. It's a very active field of research. So there we have the polymer myceles, nanospheres, dendrimers, like things like that. We have inorganic substances like silica nanoparticles, iron oxide nanoparticles, gold nanoparticles, silver nanoparticles, quantum dots, which recently they got Nobel Prize for discovering quantum dots, although it has applications in LED technology mostly, but they are also used in medicine. Then lipid-based nanoparticles. So it is liposomes, lipid nanoparticles, and emulsions. So which work better with the membranous systems. So those kind of things are there. Also, there is studies on virus-like particles, which is either taken from the viruses alone of using protein engineering, or there are different metal organic framework also that is used. So a lot of things are currently under research. So by combining nanocarriers with gene treatments such as plasmid DNA, mRNA, iRNA, miRNA, the formulation of the nanomaterial may be customized to match each patient's unique needs based on their unique genetics. Closed loop drug delivery system monitor the level of a physiological signal, such as the concentration of a molecule, and control the release as per the patient's requirement. They can work as precise dosing devices. For example, Gu and colleagues developed a closed loop system that employs integrated sensors to monitor blood sugar levels and trigger the release of insulin from pre-stock depots. This can be also applied other diseases that need real-time drug dose management. So what is a closed loop drug delivery system? Although it's pretty much futuristic, but it has been practiced. So I mean, at least tried. So if there is a pathological signal, I mean condition, there would be some signal associated with that condition. So that kind of signal would be elevated, and that would stimulate the release of drug, which would again cause the patient to come back to the normal physiological condition. And then again, this uh, uh, due to this normal physiological condition, the drug release will be inhibited. So this is a very controlled process, and also apart from closed loop delivery system, drug delivery system, these kind of drug delivery system also have the benefit of targeted therapy. So only release of medicines in a certain areas where the problem is, or in certain tissues where the disease is there. For cancer also, these kind of approaches have been used. There are a lot of papers which use this. So that's the idea. So let's come to question number 7. Dash are one class of smart polymers that can repair their structure after repeated injury and are frequently injectable by needles. Is it A, polymeric biomaterials, B, miRNA polymers, C, precision biomaterials, or D, self-healing polymers? So I'll give you a couple of minutes to think on this, and then we will be discussing the answer. Okay, so the options are pretty self-explanatory. So self-healing polymers, they are the class of smart polymers that can repair their structure after repeated injury and are frequently injectable by needles. So let's talk about smart polymers. So smart polymers are one type of soft materials that respond to environmental changes. The thermally sensitive polymers, which are frequently employed as cell carriers in 3D printing, are a good example. So self-healing polymers are one class of smart polymers that can repair their structures. So that answers our question. After repeated injury and are frequently injectable by needles. Another type of material with the capacity to remember its original shape is shape memory polymers. So that is also employed. These smart polymers can transport proteins, drugs, and cells. They can be used in precision medicine, minimally invasive surgery, and bioprinting thanks to their injectability and shape memory capabilities. So if we look at this figure, you can see that smart polymers can be used for cell therapy and personalized medicine. So smart materials are called smart because they either have the shape memory, or they are self-healing, or they can respond to certain stimuli. Like there are certain nanomaterials that, you know, they release drugs only on photothermal activation. So for them, you need to give a laser light to activate them, to excite them, and that causes also a heat based therapy, and because they get heated up very quickly, so it's photothermal therapy along with an increased drug release kinetics. So that's why these kind of materials, biomaterials, are smart materials. That's the reason. So they are packaged with containing the gene, or the stem cells, or the drugs in question, and then they are, injected into the cell. So that is one of the ways. The other thing that could be done is, in specific to, orthodontal implants, or even you know in orthopedic patients, related to bone, grafts, tissues. These kind of cells, they can be bioprinted and they can be inserted. So there are catheters, there are other kind of scaffolds which are prepared from these smart materials. So that is also one of the applications. So this figure gives us a summary of the process diagram for precision medicine, which highlights the activation time. That means the temporal control on the personalized medicine. Then there is tailored design and local delivery, that is spatial control also is there as three key principles, right? Okay, so now we come to the last question of today's session. So the term theranostics, which was first used by John Funkhauser, refers to scientific advancements that aim to create more specialized and personalized treatments for a variety of A, organoid conditions, B, animal model conditions, C, pathological conditions, or D, humanoid conditions. So I'll be giving you a couple of minutes to think on this, and then we'll discuss the answer. Okay, so the term theranostics, which was first used by John Funkhauser, refers to scientific advancements that aim to create more specialized and personalized treatments for a variety of pathological conditions. So let's see what we have about theranostics. So theranostics simply means a combination of therapeutics and diagnostics. So that's a combined term for these two. The term theranostics, which was first used by John Funkhauser, refers to scientific advancements that aim to create more specialized and personalized treatments for a variety of pathological conditions. These advancements also aim to combine diagnostic and therapeutic applications into a single agent, resulting in a promising therapeutic paradigm that includes diagnosis, drug delivery, and treatment response monitoring. It provides a diagnostic therapy for specific patient, testing them for potential adverse drug reactions, and then customizing a course of action for them depending on the test results. So this is a schematic for theranostic applications. So formulation of the design, micro and nano engineering and delivery mechanisms is important for the therapeutic approaches, also for diagnosis, as a matter of fact. And then this could be applied for biomedical imaging and therapy. So as I talked about the photothermal therapy already, then there is non-invasive and highly sensitive disease, early diagnosis and surveillance. Then there is nanotoxicology, environmental nanoparticles, and disease development. So all these are applications. So with that, we are at the end of week 10th problem solving session. So just for everybody, if somebody has missed something, we will be going back to going through the questions once again. And after that, if you have any questions, you can ask that, or we can call it a day. So let's see, let's go to question number 1. So question number 1 was Ayurveda. It classifies all individuals into different prakriti based on the theory of three dosha that includes vata, pitta, and kapha. So that's the answer. So Ayurveda categorizes drugs according to Ras Panchaka, that is Ayurvedic pharmacology, which states that the drug's action is attributed to certain factors present in the drug as follows. The wrong combination here is guna, the effect. So it would be rather gun or property. Okay, so Garrett is best known for his book, Inborn Errors of Metabolism, where he claimed that 4 diseases, alkaptonuria, albinism, cystinuria, and pentosuria, were inherited as Mendelian autosomal recessive trait, and was published in the year 1909. Okay, coming to the next question, in 1926, Palmaquine, an 8-aminoquinoline, was one of the most effective medications used to treat acute malaria, and it had a adverse effect of deficient of basically anemia, hemolytic anemia, and that led to the advent of personalized medicine. Anyway, Shendure's group published the first proof of concept that next generation sequencing technology may be used to diagnose genetic diseases in September 2009. After a few months, they reported Miller syndrome, the first recessive disorder through whole exome sequencing, where all the exons are sequenced. We talked at length about next generation sequencing technologies, and then about the limitations as well. So coming to the next question, engineering smart biomaterials for drug delivery systems that can detect the demands of a person's physiological condition and change their drug release profile accordingly is an appealing method for creating precision pharmacokinetics. So that's the answer. We discussed about pharmacokinetics as well. And then we talked about the question number 7, that self-healing polymers are one class of smart polymers that can repair the structure after repeated injury and are frequently injectable by needles. So we talked about smart polymers, which can remember their shape, which can, of course, self-heal, or they have some potential other applications then coming to the last question, the term theranostics was used first by John Funkhauser, refers to a scientific advancement that aimed to create more specialized and personalized treatments for a variety of pathological conditions. So with that question, we are at the end of today's session. If there are any questions, you may ask them now. If there are none, then we may call it a session today, and then we will be meeting back uh, on next Tuesday for the week 12 of this course. So we'll be meeting next Tuesday at the same time, 6 p.m. to 8 p.m. And we will be discussing about the ethical concerns of genome engineering. Basically, that's what is there in the last week. So yeah, all the best then. Um, I'll stop the recording.